Welcome back to a familiar course in an unfamiliar setting. We're going to take Chapter 21 right from the top since it's been a while. Please remember to post questions to the forum on Moodle as you have them, and I'll answer them for everyone there. As a reminder, these intro slides are a good high-level overview of what we are learning, so you can always refer back to them. Also remember that you have reading guides and lecture notes posted in Moodle, and you'll get the most out of these videos if you've done the reading as assigned. We're talking about enzymes in Chapter 21, and remember that enzymes are a form that proteins take, one out of many forms that the proteins take. And because vitamins are often associated with enzyme functions, we'll talk about vitamins in this section as well. Enzymes are any organic compounds that act as catalysts for a reaction, and most of the time they're proteins. We'll talk about a few special examples in Chapter 22 where ribosomes function as enzymes. Some of the nomenclature that we had talked about in proteins in Chapter 20 is a little bit different here in Chapter 21. So in Chapter 20, when we talked about protein classification, we might have talked about monomeric or multimeric proteins, monomeric being just one polypeptide chain and multimeric being multiple peptide chains. And then we talked about simple versus conjugated, simple being only amino acids and conjugated having something other than amino acids, a prosthetic group. We use those same terms to refer to enzymes, but we also get a little bit more specific when we talk about the prosthetic groups. The prosthetic groups for enzymes are called cofactors when we're talking about enzymes. So a cofactor is a specific example of a prosthetic group. When we talk about an enzyme then that has the cofactor, the enzyme with the cofactor is called the holoenzyme, think like whole enzyme, and the enzyme without the cofactor is the apoenzyme. We can also further narrow down cofactors so that cofactors can be designated as inorganic or organic. The cofactors that are inorganic we still will call a cofactor. Some really common examples are like magnesium, which is responsible as a cofactor for many enzymes that deal with DNA. If cofactors are organic molecules, we call it a coenzyme, and most vitamins are coenzymes, and so this is something that we'll spend some time talking about at the end of this chapter. When we name enzymes, their names will either end in "ase" or in "in". Most commonly, enzyme names end in "ase". That refers to the fact that they have catalytic activity. Some enzymes will end in "in", especially if they were discovered long ago. So if you see a word that ends in "ase" or "in", you should know that it's an enzyme. The enzyme name might be made up of one or two words. If the enzyme name is made up of two words, the first word always indicates the substrate. That's what the enzyme is acting on. The second word will always indicate what type of reaction is happening on that substrate. If the enzyme is made up of only one word for its name, the reaction is always hydrolysis, and the substrate is just going to be the prefix. So for example, we should be able to identify what each of the following enzymes is doing, at least in general. So the first enzyme, cellobiase, is just one word, so we know that it's going to hydrolyze, and it's going to hydrolyze something that's beginning sounds something like cellobi. We know the molecule cellobiose. Cellobiase hydrolyzes cellobiose. This works whether or not the enzyme is actually something you know, or if it's something completely unique. So for example, textbook disintegrase, the reaction would be a disintegration. It might disintegrate your textbook. We don't actually need to know anything about the enzyme to know what its function is. We only need to know its name. While many of the reactions that we need to identify are reactions that we talked about all the way back in organic chemistry, it's helpful to have a reminder of the types of scopes that enzymes can have and that you'll see in their names. So for example, we have 6 major classes of enzyme nomenclature, each of those having subclasses that refer to specific reactivity as well. The first class is oxidoreductases. They're responsible for oxidation and reduction. Transferases are responsible for moving a group from one place to another. Hydrolases are responsible for adding water to break things into pieces. Lyases are oftentimes responsible for forming double bonds, either by removal of water or removal of carbon dioxide. Isomerases will change a molecule from one isomer to another isomer, and ligases are going to join two pieces together to make a new, larger molecule. Almost always these require energy, and almost always these require ATP. We absolutely need to know the 6 major classes of enzymes, but within those classes, it's also important to know certain subcategories that occur very often biochemically. So again, the first class of compounds, of enzymes that we need to recognize are oxidoreductases. Oxidoreductases perform redox reactions, which we talked about in chapter 9.3 of Gen Chem, but we also talked about it in chapter 14. Primary and secondary alcohols can be oxidized. We talked about it in chapter 15. Aldehydes can be oxidized, ketones cannot. And we talked about in chapter 16 as a way that carboxylic acids are produced. Generally, we see increases in the number of bonds to oxygen or decreases in the number of bonds to oxygen will indicate an oxidation or a reduction. We also see that oxidations and reductions occur when we move between an alkane, a single bond between carbons, and an alkene. Transferases are a second class where we move a functional group from one molecule to another. Two types of transferases that occur very often in biology are transaminases, which are responsible for moving amines and processing amino acids in our amino acid metabolism. Again, transaminases are really important to remember because of their central role in amino acid metabolism. And kinases are really important because they move phosphate groups. Now, a kinase is a transferase that moves phosphate groups, which means the phosphate group is moving from one molecule to another molecule. That molecule might be ATP, or it might be any other molecule. Hydrolases are performing hydrolysis, where we add water to break a large molecule down into smaller molecules. That can happen with our 3 major biological molecules: carbohydrates, lipids, and proteins or peptides. When we add water to break down a carbohydrate, it will break the acetal or the glycoside bond, and we call those enzymes carbohydrases. When we add water to a lipid, that will break an ester bond for our saponifiable lipids, and we call those types of enzymes that perform that reaction lipases. And when we add water to a protein or peptide, it will break the amide bond that forms the primary structure of that protein or peptide, and we call those proteases. We also have lyases. Lyases are perform addition or elimination reactions. Think of the reactions that we talked about with chapter 13 as addition reactions. They are either adding on to a double bond or they're eliminating to form a double bond. Usually we're talking about adding on water or adding on an amine, or eliminating water or an amine. We also sometimes will eliminate carbon dioxide in lyases to form an amine. Isomerases are going to perform reactions that yield an isomer of the original product, so it's going to have the same formula, but it will have a different structure. These are fairly common in our central metabolism. And ligases are going to perform reactions that combine 2 molecules in one. Notice that ligases require ATP as an energy source, but the phosphate is not transferred onto any of the molecules. Only the energy to break ATP apart is used to combine those molecules into one. Shown on the screen are several examples of reactions that you should be able to look at and identify what type of reaction is occurring, which major class of reactions is at play, or which major class of enzymes is at play. So if we look at the first reaction, we can see that we have an alcohol at this location that is becoming a ketone at that same location in the product. Because an alcohol only has a single bond to oxygen, and now that same location has a double bond to oxygen, we know that we have an oxidoreductase as the enzyme at work here. In the second reaction, we see that we're adding water to a molecule to break it into 2 smaller pieces. What are those pieces? Well, each of those pieces has an amine, and then we have a carbon, and we have a carboxylic acid. So each of those pieces is an amino acid. We are taking a group of amino acids that are linked together and making 2 individual amino acids. This type of reaction is catalyzed by a hydrolase, adding water to break things into pieces, specifically a protease, where we take a protein, a polymer of amino acids, and break it down into its amino acids. The next reaction has a fructose 6-phosphate, which becomes a fructose 1,6-bisphosphate. So we're moving a phosphate onto the molecule. You can see on the fructose 6-phosphate, this alcohol is free, and on the fructose 1,6-bisphosphate, we've now hooked a phosphate onto that alcohol position. That phosphate came from ATP. That doesn't mean that we're spending the ATP just to join the molecules together. We're actually moving the phosphate onto the molecule, which is why this is classified as a transferase as opposed to a ligase. The final molecule is going to eliminate water and form a double bond. So in this final reaction, we have the elimination of water and the formation of a double bond. When we have a double bond formed by eliminating water, or when we add water to a double bond, we should think of a lyase always being the type of enzyme at work. We have several models of enzyme activity that can help us to understand what's going on in the enzyme itself. The first of those is the lock and key model, which we can see in the top row. We see that substrate can bind to an active site that's perfectly formed to fit that substrate. Once it's bound, it forms what we call an enzyme substrate complex, and that enzyme substrate complex can catalyze the conversion of the substrate into a product or multiple products. We also can talk about a model where the enzyme active site doesn't exactly fit the substrate, but it's pretty close. Once the enzyme and substrate bind together and we have the enzyme substrate complex, the enzyme substrate complex has some amount of strain that helps to catalyze a conversion to products. That's called the induced fit. Some models apply better to some enzymes and others to other enzymes, but in reality the truth is somewhere in between. We have not exactly a perfect fit, but it's very close in most cases. The thing that is consistent between both of these models is that both models require an active site. That's the area of an enzyme where the substrate binds and the reaction takes place. If we do not have an active site, we cannot have any sort of enzyme activity. The reaction rate will be almost nothing. And so all of the types of things that unfold proteins will also unfold enzymes, since enzymes tend to be proteins. And when those proteins unfold or are denatured, when the enzymes are denatured, we see that there is no active site and no enzyme activity. Enzyme active sites will be formed by the three-dimensional structure of the enzyme protein itself. The three-dimensional structure will have an active site that has R group interactions or side chain interactions that are tailored to match the substrate. By tailoring these interactions to match the substrate to different levels, the enzyme is able to deliver different amounts of specificity. Some enzymes will be absolutely specific or absolutely selective. Absolutely selective enzymes will react on exactly one substrate only. While this is relatively rare, it's actually really important for our central metabolism because we want our central metabolism to perform specifically one reaction without very many side reactions. We also might have stereochemical selectivity. In stereochemical selectivity, we will only react one set of stereoisomers, but not the other. So for example, we might have an enzyme that can react on all carbohydrates that are D, but no carbohydrates that are L. This allows that enzyme to have a wide-ranging scope of action, but it will only react on the naturally occurring carbohydrates. An example of this is alpha amylase, which is the enzyme in our spit and in our intestines that breaks down polysaccharides. It can react on any length of starch, but it will only react on the D carbohydrates, on D amylose. We can also see group specificity. Group specificity is where we only react on one set of functional groups, so like amino acids, or linkage specificity where we only have one type of bond. So we might have acetyl functional groups that are not glycoside bonds that can't be reacted by an enzyme with linkage specificity, but they might react with glycoside bonds specifically. Enzymes not only have selectivity for different types of reagents, they also have varying levels of activity depending on what type of environment that enzyme is in. There are four major factors that affect the rate of enzyme activity, the first of which is temperature, which we can see displayed on the left. We notice that as temperature increases, initially we're going to increase the reaction rate in this part of the graph. But once we reach an optimum temperature, then we switch and we end up having a decrease in enzyme reaction rate. When we're increasing the reaction rate, what's shown in yellow, that's usually because we're increasing the overall kinetic energy. Remember that for a reaction to occur, it requires us to have a certain amount of energy, and the higher the fraction of molecules that have sufficient energy, the faster the reaction occurs. So when temperature increases, more reactions occur because there are more molecules with sufficient energy. However, as temperature continues to increase, we meet a point where enzymes begin to denature, they begin to unfold. And when enzymes unfold, they no longer have an active site. Without an active site, they can no longer catalyze the reaction, and so we see denaturation will start to cause enzyme rates to go down because the enzymes are no longer active, what's shown in red. The optimum temperature can be different depending on which enzyme we're talking about. Most often, those enzyme optimum temperatures match the environment the enzyme is likely to be in. So for humans, most of our enzymes are optimized for 37 degrees Celsius or 98.6 Fahrenheit because that is the environment that our enzymes work in. We're going to do an enzyme lab, which we won't do anymore, where we have potatoes. The potatoes enzymes are optimized to be most active at temperatures that potatoes are likely to encounter, like around maybe 30 degrees Fahrenheit to 70 degrees Fahrenheit soil temperature. We also see that pH will affect the enzyme rate. pH will affect the enzyme rate going down from an optimum pH on either side. So if we have an enzyme that is below the optimum pH, the increase in acidity, lower pHs, higher acidity, the increase in acidity will protonate more of the side chains and cause the protein to unfold or denature. The same thing happens if we decrease the acidity or increase the pH. The protein begins to unfold, denature, and we no longer have an active site. So there's some optimum pH where the protein remains folded, the enzyme remains folded, and we have a maximum rate. Again, this maximum rate is going to match the type of environment that the enzyme is likely to be in. So the maximum rate pH is going to be around 1 for our stomachs that have a pH of around 1, but since our bloodstream is around 7.4 for a pH, most of the enzymes in our bloodstream are optimized for a pH of 7.4. We also can see a change in rate as we increase the amount of substrate concentration, which is the next graph over. Notice that as substrate concentration increases, at the beginning, we see the reaction rate go up very drastically. But as we continue to increase substrate more and more, the reaction rate levels out until it approaches some maximum reaction rate. We can never go faster than the maximum reaction rate. This is the maximum theoretical efficiency for that enzyme. We can think of this kind of like a factory worker. A factory worker can only work at some amount of maximum rate. If the factory worker has all the supplies the worker needs, the worker can work close to that maximum rate. If the factory worker does not have all the supplies it needs, if the substrate is low, then the reaction rate will be significantly less. And as substrate becomes available, as materials for the factory worker become available, the reaction rate quickly increases towards that maximum rate. And of course, we can also see that reaction rate will increase as we increase the enzyme concentration. We can use the same factory worker example to talk about this. If we have more enzyme, it's like having more factory workers. And the more workers that you have, the faster they can work.